This is Module 5, Carbohydrates and Lipids. In these next two videos, I'll be describing lipids specifically. In this video, I'll be talking about the structure of different types of lipids, either energy storage lipids or membrane lipids, structural lipids, and also their function, so energy storage or structure. Lipids are hydrophobic molecules and are not able to form hydrogen bonds with water. Because of the hydrophobic effect, lipids in an aqueous environment tend to cluster together away from water in order to reduce the amount of ordered water that tries to form around it. And so the general characteristics of all lipids are that they are hydrophobic, they are predominantly hydrocarbon based, and so remember that hydrocarbons are not able to form hydrogen bonds with water. They are also insoluble in water. However, there are some lipids, particularly structural lipids, that have polar components that allow them to form membranes in water. More on membranes in the next video. The major functions of lipids are structure and energy storage, and those are the two most major functions. There are additional functions that where lipids participate in cell recognition and cell-to-cell signaling, but I'm not going to be describing them this semester. But so the major functions that we have to worry about are that lipids are the major structural component of all cell membranes, and they are also representative of high energy storage molecules, and you can create a lot of ATP using lipids as starting material. Lipids are often sort of interchangeably used with the term fatty acid. So lipids and fatty acids are the same. Fatty acids are carboxylic acids with very long hydrocarbon side chains of various lengths. And so these are three examples of three different fatty acids. These are the two most common fatty acids, palmitic acid and linoleic acid. Palmitic acid is the major source of oil in palm trees. It's also found in meats, cheese, butter, dairy, and chocolate. It's added to shampoos and cosmetics and is sometimes used as a cleaning agent. And any excess carbohydrates in your body are rearranged and packaged as palmitic acid. Palm oil is a pretty sad story. If you look into it, do an internet deep dive on palm oil. But it's the forestation of and creation of palm oil is responsible for 8% of the world's deforestation. And so there's lots of clear cutting for production of palm oil. Just so sad because it's actually one of the most common fatty acids. Another type of oil, linoleic oil or linoleic acid, is a type of polyunsaturated oil. Oh, I forgot to mention, palmitic acid is a saturated fat, meaning that all of the palmitic oil, is that picture here? Yeah, sorry. So all of the fatty acid chain in a saturated fatty acid contains no double bonds. So there's no double bonds within the long fatty acid chain of a saturated fatty acid. An unsaturated fatty acid, like linoleic acid, contains double bonds. And in fact, linoleic acid is considered a polyunsaturated fatty acid, meaning it contains two or more double bonds. And the presence of these double bonds will affect how these lipids pack together tightly in an aqueous environment and help to form the structure of a membrane. Linoleic acid linoleic acid is one of your essential fatty acids. So it is the omega-6 fatty acid. It's often found in beauty products and things like oil-based paints. It's also a really major component of plant oils. Without linoleic acid, so like I said, it's an omega-6 essential fatty acid. You have to get this from your diet. And so if you don't consume enough linoleic acid in your diet, you end up with hair loss, the inability to repair wounds, skin wounds, and also skin scaling, where your skin appears very scaly. Okay, so the three major types of acids, fatty acids, are saturated fatty acids, where there's no double bonds, unsaturated fatty acids, where there's at least one, and then polyunsaturated fatty acids, where there's two or more double bonds. This is actually related to how we name fatty acids based on the placement of double bonds and also specified based on the chain length of the hydrocarbon chain. And so we put the carboxyl group of the fatty acid up at the top and label that 1. And then you can count how many hydrocarbons there based on counting the number of carbons. And so for oleic acid, which is an unsaturated fatty acid containing 1 double bond, there are 18 carbons in oleic acid. So 18 carbons starting with the carboxyl at the top and ending with this one at the bottom. And then you place, so 18 specifies how long it is, and then :1 specifies that it has a single double bond. And then this designation here tells you where the double bond begins. And so you would put the term delta 9 to specify that the double bond begins at the 9th carbon. Just as a side note, oleic acid is found in both animals and plants. It's also used as a soap, as an emulsifying agent for mixing insoluble things with water. Okay, linoleic acid, however, contains 2 double bonds. So this is a polyunsaturated fat. It also has 18 carbons, starting with the carboxylic acid at the top. And then it has a double bond starting at the 9th carbon and also at the 12th carbon. And so you would designate how many double bonds it has after the colon, and then the location of where the double bond begins. And then you repeat that sort of type of naming system with other types of fatty acids containing 3 or more double bonds. The double bond matters. The double bond will basically dictate how tightly packed these fatty acids can get. So saturated fatty acids, which contain no double bonds, pack very tightly together. And I'll describe more about how this works in a future slide. Unsaturated fatty acids that contain double bonds will take on this cis configuration where there's like a bump, a kink in the hydrocarbon tail. And that's because of the double bond. There's no rotational freedom around that bond. And so we end up with this bend. A trans fatty acid is a fatty acid that contains a double bond, but is in the transformation. And so there's no rotation around this either because of this double bond. And these trans fatty acids are artificially generated. You don't typically find high amounts of trans fatty acids from a natural source. There are some very small amounts, but not the amounts that you would typically see in like fried foods and things like that. The degree of saturation, so how many double bonds you have and where they are, will affect the ability of these lipids to pack tightly together. And so as I was stating on the previous slide, saturated fatty acids pack very tightly together because they don't have a double bond, and so they're able to line up end to end with no bumps or kinks or movement in the middle. And so the reason why these pack so tightly together is because all of the atoms that are lined along the length of the hydrocarbon are in van der Waals contact with neighboring molecules. And so those van der Waals interactions, the cumulative sum of all of those van der Waals interactions located all along the hydrocarbon tails stabilizes this formation, and these fats are solid at room temperature. They're not very pliable. They're not very flexible. However, in an unsaturated fatty acid that have a double bond, and this bend forms because of the lack of rotational freedom around that double bond, this introduces this bend, which interferes with the ability of these van der Waals interactions to take place. And so these are not in van der Waals contact, and we increase the fluidity as we increase the amount of double bonds in the tail. And so this creates a lot of instability in the ability for these molecules to pack very tightly together. And so this actually is a more fluid structure. It's not solid at room temperature, it's liquid at room temperature. I just want to take a moment to discuss trans fatty acids because this does come up each year. Trans fatty acids are created by pumping hydrogen molecules into saturated fatty acids to create double bonds in a transformation. And so and unsaturated vegetable oils. So unsaturated fatty acids is how it starts. And then we pump hydrogen molecules into this one to change the configuration of this bond to a trans double bond. And so the cis bond moves from cis to trans, and now these can pack very tightly together and are solid at room temperature. They are very long-lasting types of fats, cooking oils. They have a very long stable and safe shelf life. They're very, very cheap to manufacture. And so they were used pretty ubiquitously in American cooking when they were first introduced. Crisco is an example of this. So it's shortening. It sits on the shelf for years and years, never loses its taste, and never really goes bad. But trans double bonds and trans fatty acids have been associated with adverse health effects. So for example, if you consume too many trans fatty acids, this increases your risk of heart disease. It increases the amount of fatty acids floating around in your blood, and it also increases the amount of bad cholesterol floating around in your blood. Both of these things will increase your risk of type 2 diabetes and inflammation of the heart and the gut. And so it's not totally clear what the mechanism is behind this, but with increasing consumption of trans fatty acids, you get the increased risk of these really bad health effects. And as a result, many companies have reduced their use of trans fatty acids, and in some states, the use of trans fatty acids in fast food development is banned. Okay, now let's start to take a look at the structure of lipids and how they relate to their function. There are two major functions for lipids: storage, energy storage, and some type of membrane structure, a membrane lipid. So all of these lipids here are membrane lipids, and this one lipid belongs to this category of storage. Lipids are categorized based on their structure, and there are three different types of structure that the lipids are characterized based on. The structure of the backbone, so whether it's a glycerol backbone, as pictured here, or a sphingosine backbone, which we'll talk about the structure of that in a moment. So first, the backbone differs. Then the constituents, what's actually attached to this backbone, can differ. And so the constituents are there fatty acid chains? Is there a head group? And what is the nature of that head group? Is a phosphate head group or maybe a saccharide head group? And so the structure of these lipids differs based on what's attached to the backbone. And then we can also classify them based on the type of linkage that exists between the backbone and the constituents. So whether or not it's an ester linkage between carbohydrate groups, or whether or not it's an amide linkage between amino groups. I'm going to show you examples of all of these different types of things. So this is a sphingosine backbone. These are the different constituents. They can be fatty acid chains or head groups, and the nature of those head groups can be different depending on if it's a phosphate group or some kind of sugar base. They're also, I just want to point out that membrane lipids tend to be polar because they have polar components in their head groups. Their head groups can be polar and interact favorably with water, and then the non-polar components are the fatty acid chains, whereas storage lipids tend to be more neutral because they're mostly consisting of non-polar fatty acid chains. Let's first take a look at storage lipids. There's only one type of storage lipid. It's a triacylglycerol, commonly referred to as a TAG. Triacylglycerols are composed of a glycerol backbone, which is pictured here, and the constituents are three fatty acid chains, which can vary in length, all linked to the glycerol backbone by an ester linkage. And so if we were going to categorize this based on these three components, 1, the backbone is glycerol. 2, there are 3 fatty acid components. And 3, those components are linked to the backbone by an ester linkage pictured here. These are non-polar fatty acids and hydrophobic in nature, so they will cluster together when put in an aqueous environment. And as such, tags are stored in adipose tissue in the body, all clustered together away from the aqueous environment of the interstitial fluid. Tags, when hydrolyzed, can yield more energy per unit than carbohydrates are. And so we typically store tags because they represent such a high energy molecule. Stored fats can supply nutrition during starvation for about 2 to 3 months. You can access your stored fats, whereas stored sugar, which takes the form of glycogen, provides much fewer, only about 2,000 calories of nutrition, and will basically be used up. All of your stored carbohydrate will be used up within the first day or 2 of starvation, really the first day of starvation. After you use up all of your glycogen, then stored fats are accessed for energy production. That's the storage lipid, that's it. All of the rest of this is going to be about structural lipids, or those that make up different components of membranes. These are the different types of structural lipids that you will find in plasma membrane bilayers, and I'll talk more about the plasma membrane and how it comes together in the next video. These molecules are amphiphilic, meaning they have both polar components and nonpolar components. And those polar or hydrophilic components can vary in complexity, and they are these blue components here. They are the head groups attached to the different backbones. Let's start with the glycerophospholipids, which are pictured here. There's two major types of membrane lipids. There's the phospholipids and the glycolipids. Phospholipids have a phosphate head group. Glycolipids have some type of carbohydrate head group. So first, let's talk about phospholipids. Glycerophospholipids have 1, a glycerol backbone, 2, 3 different components, 2 fatty acid chains, and a head group that is typically polar. And then the polar head group is linked to the glycerol backbone by a phosphodiester bond. The fatty acids are linked by ester linkages, as pictured here. The fatty acid, in this glycerol phospholipid, the glycerol backbone is pictured in black. The 2 fatty acid chains are pictured in red. And the head group, the phospholipid head group, is pictured in blue. These head groups are polar or charged head groups pictured in green. So this part is linked to the blue part by a phosphodiester linkage pictured here. The name of each of these different types of glycerophospholipids comes from the specificity of the head group. And so I'll talk more about that on the next slide. Again, these are amphiphilic or also amphipathic molecules. This is a synonym. Amphiphilic and amphipathic mean the same thing, which means they contain both polar and nonpolar components. These are one of the major components in your lipid bilayers. So this is the major component in lipid bilayers in cell membranes. Sphingolipids, so another type of phospholipids. So this is that chart was at the beginning, the structural lipid chart. So now we're still talking about phospholipids. But now if your components of the lipid are linked to a sphingosine backbone, we collectively refer to them as sphingolipids. A sphingosine backbone is one that looks like this, where one of the positions that would normally be reserved for attaching a head group actually has a chain, a hydrocarbon chain attached to it. And so compare that to this glycerol backbone, where here we can actually add a component, a constituent, but here we can't because it's contained within the backbone of the sphingosine. And so the sphingosine starts out as a glycerol backbone. It looks like a glycerol, except at the center carbon, there's an amine group. And then at this carbon, we have a long hydrocarbon chain that is the backbone. So you can't cleave that off and use it for anything. The component that's linked to this sphingolipids in the center is the fatty acid chain. And then we also have a head group. And the nature of the head group can be different. It can be a phosphate-derived head group or a sugar-derived head group. Once we attach so the sphingosine is referring specifically to the backbone. Once we start attaching things like components to the sphingosine backbone, we call it a ceramide. And then the ceramides can change depending on the type of head group you add and so on. And so there's a different head group for different ceramides. I highly recommend you create a chart or flow chart or some kind of whiteboard to keep all of these categorized properly in your head. Okay, and I'm going to summarize them in just a moment too. So now we're looking at sphingolipids. So you can have 2 different types of sphingolipids, one that has a phosphate head group and one that has a sugar head group. So again, one that has a phosphate head group or a sugar head group. Sphingolipids are ones that have a phosphate head group. One type of sphingolipid is a Sphingomyelin. And this has what's called a phosphocholine head group, which is a phosphate group attached to a choline group, which is this nitrogen attached to different methyl groups. Sphingomyelin I'm going to showcase in just a moment. So that is a specific type of sphingophospholipid. Again, it has a sphingosine backbone and a phosphate head group. If it's attached to a choline group, we call it a Sphingomyelin. And then there are other types of sphingolipids that are categorized under glycolipids because they have some type of mono, di, or polysaccharide attached as their head group. Here are 3 different types of glycosphingolipids, and they are different depending on the type of head group they have. For example, this one has a glucose head group, this one has a di, tri, or tetrasaccharide head group, and this one, the ganglioside, has a very complex oligosaccharide as its head group. And so we can categorize different glycosphingolipids based on the nature of their carbohydrate head group. So let's look at kind of all of those lined up so we can see some differences all on one page. Here's the backbone, the sphingosphene backbone. Here's where we can put a head group. Here's where we can put a constituent like a fatty acid chain. Oh, sorry, no, here's where we can put the fatty acid chain, sorry. And a sphingosphene starts out as a glycerol backbone, but instead of having an OH group here, it's got this amine group here. And so fatty acid chains are linked to a sphingosphene backbone by an amide linkage. And once we add that fatty acid chain, we now call it a ceramide. And so all of the rest of these fatty acids are derivatives of a ceramide. They all have this chain attached to that amino group. Now let's start adding different types of head groups and see how that changes the categorization. So here's a Sphingomyelin. So it starts with a sphingosphene backbone, just like this. We attach a fatty acid, so now it becomes a ceramide. And then we attach what's called a phosphocholine head group. It's got a phosphate head attached to a choline group, which is a nitrogen attached to methyl groups. So that makes it specifically a Sphingomyelin. This is another type of Sphingomyelin that has a different type of choline head group. But both are called Sphingomyelins because they both have some form of this choline head group and phosphate head group. The Cerebrosides, again, they start up as a ceramide, right? They're a sphingosphene backbone, pictured here, attached to this amine group as a fatty acid residue, so now it's a ceramide. And then we attach a single sugar residue as its head group. And so this sugar residue makes it a glycolipid, a glycosphingolipid. And now let's look at the ganglioside. This is where things get weird and complicated. So here's our sphingosphene backbone. There's the ceramide, because we've linked a fatty acid chain via an amide linkage. And then this whole thing is the head group, this really complex group of oligosaccharides attached to a sialic acid makes this a ganglioside. And so you can see they all share the same backbone. They all share the same amide ceramide linkage. The only way in which they really differ is by the nature of their head group. These have phosphate head groups, and so they would be classified as Sphingophospholipids. These have carbohydrate head groups, and so they would be classified as glycosphingolipids. Let's look at sphingomyelin more closely because this is a really important structural component of nerve cells. Sphingomyelin is a ceramide with a phosphocholine head group, and so it has a sphingosine backbone, a fatty acid chain linked to it by an amide group, and a phosphocholine head group, which is a phosphate group attached to this choline nitrogen group, which is a nitrogen attached to different methyl groups. Sphingomyelin is the major component that makes myelin, the myelin sheath of your nerve cells. And so all of your axons are, the length of the axon is protected by the myelin sheath. And the myelin sheath is hydrophobic. It's a fatty component protecting the nerve from damage, and it also helps to helps the nerve to fire its synapse. And so charges race down the axon much faster than they would if they didn't have this myelin sheath. And so the myelin sheath is composed of sphingomyelins. Cerebrosides and gangliosides are also ceramide derivatives, right? They have the sphingosine backbone. They have a fatty acid chain attached by this amide linkage, so that makes it a ceramide. And then cerebrosides and gangliosides differ based on the components of their head groups. A cerebroside, which is present in oligodendrocytes, these produce myelin for the central nervous system, and they're found in the brain. These have a single monosaccharide unit as its head group. However, gangliosides have this really complex oligosaccharide head group attached to the sphingosphene backbone, and this is actually a ceramide derivative. Note the ceramide linkage right here. Gangliosides are really important for signaling, and the reason why they're good at signaling has to do with the complexity of this big head group. They're also really important for identifying cells as self or not self, because these are unique combinations of polysaccharides that tell your cells that that cell is supposed to be there. This one is largely found on the outer face of plasma membranes and will accumulate in people who have Tay-Sachs disease. Tay-Sachs disease is a lipid disorder where you're not able to degrade and recycle these lipids, and so they build up and cause problems. People with Tay-Sachs disease tend to die before they're 4 years old. So what do you need to know from all of this? You need to know the different types of classifications and what they're based on. So is it a, you need to know if it is a sphingolipid or if it's a glycolipid. You need to know if it's a, you need to know if it's a glycolipid or a phospholipid. You need to be able to distinguish sphingolipids from glycerophospholipids. And you also need to remember that there's a tag in there, the triacylglycerols. You need to know the components of each types of those and the different backbones, the glycerol backbone or the sphingosine backbone. And you should be able to recognize and identify the structures, but I will not require you to draw them.